think what I'm going to do, I just didn't really know what I would cover, but I decided to try to give a general perspective on some of the things that I feel are very interesting in the field of sleep and sleep disorder medicine. It will be a little bit more concentrated on human research, but that doesn't mean that I don't think that animal research is important. I think in general, we all agree that what we heard, that one of the exciting things about sleep research is that there is still a big unknown. We know quite a bit about the molecular basis of the circadian clock, but the molecular basis of sleep homeostasis, of sleep debt, is really completely unknown. And I think that's what we started to see being approached with some of the animal work and across species, etc. The second thing that we heard is that actually a lot of the population has sleep problem. And unfortunately, I think that's often a problem of a lot of the studies we see, is often people see only one side of the animals. So people, for example, are interested in sleep apnea. We have heard the complexity of the phenotype of sleep apnea through Danny Aker, for example. But very often people who study sleep, they just completely ignore, they don't even record breathing. And honestly, we know that I'm sure I have some sleep apnea. Everyone has sleep apnea past a certain age. So ignoring sleep apnea, you can't really do good sleep study. And I think it's the same for most of these other disorders that are very, very common, including circadian dysregulation. Right now, we really don't know. Sometimes people are tired during the day, but we don't know if it's because their sleep is not good enough or if their circadian clock is completely off target. And then, of course, it has a large economic impact. So I think what's really cool about sleep, in my opinion, is that it's at the confluence of many revolutions. You know, I think I mentioned, of course, we talked a little bit about in models and all the really exciting works that's being easier and easier that can involve even new molecular models, like we heard about jellyfish. I mean, now you can sequence the jellyfish genome quite fast, so it's very exciting. There's definitely also a genomic, proteomic revolution, especially the genetics. We heard about the UK Biobank. It's amazing. You can get this very, very large sample and get an idea of what is really associated with specific traits. And I will also argue at the end, and I think Aditya in one of the next talk will talk more about proteomic, which I think is a very, very also promising approach. And also there is a complete revolution in hardware and software, which I think we hear a little bit more, which I think is just starting to be applied to sleep. And with all this is what we do. And that's really sad. I mean, I'm sorry to say, because this is like a very old technique. People are all looked up with all these cannulas. They measure really like your EEG waves. They measure your breathing because sleep apnea is so frequent. They measure your leg movement. They measure your snore. And then people do an interpretation, and actually technicians just look at it, and they move 30 seconds by 30 seconds, and they decide what kind of sleep stage you have, if you stop breathing, if you have leg movements. It's totally primitive. It's just amazing that we still do this. And then we measure the sleep cycle and the sleep apnea. And really at the end, we only get like 2 or 3 numbers. The number of time people kick their legs during sleep, how sleep is consolidated, how many sleep apnea events you have. And again, we have heard from the previous talks how complex each of those things are. That sleep apnea is not just one disease, it's not just EHI. We have heard that restless leg, PLMI, is actually very complicated. Insomnia is very complicated. So we're really completely, I think, a little bit in the dark. And my whole point here is, I think we don't use the data we have. I don't know if you realize, but there's about 1 million sleep studies that's conducted every year in the US. And this is recording all these things that we have now, the EEGs, the EMG, the breathing, etc. And all that data is thrown away. I mean, we just summarize it by 2 or 3 numbers. So I think it's a huge opportunity that's being missed. So when I started to look at this, I kind of identified a couple of things that I think were missing. One was really to create like a single online questionnaire that would cover all sleep disorders. Again, one of the problems we have is sleep medicine is very, discipline specific. So if people study sleep apnea, they have their sleep apnea questionnaire. People study RLS, they're neurologists, they have their RLS questionnaire. But if people study insomnia, they have their own questionnaires. But it's quite difficult to bring everyone from this different discipline together, which I think we have the luck of having at Stanford because we have a very multidisciplinary center, and try to really cover all these disorders through this validated scale in one questionnaire. And we now use this all in our sleep clinic. a second thing that I thought was very important. So it's good phenotyping, but subjective, because people won't complain about having too many spindles. They will complain about insomnia, and actually that's what matters in treating patients. Then the second area that's very clear is we still have human technicians that score sleep. It's totally outdated. It doesn't make any sense with the new machine learning, and I will tell you a little bit about the work we have done here. Then I think I won't talk too much about this because I think it will be covered very nicely by our next speaker, Jonathan Berendt, but there is a need for better system. I mean, this PSG is on a death row. And in fact, I think there will probably be systems that can record sleep at home, and then we can measure all these things continuously, and even as predictor of disease, et cetera. So that's very exciting. And finally, there is a need for biological markers. The circadian field in particular is really a pity because we have this very, very strong biology and genetics, and we know the mechanism, but yet we still don't know how to measure the circadian clock properly. And I think Aditya will also talk about this. And then finally, I think if we're at home, we can probably do intervention directly at home. So hopefully the field, I think, will change completely in the next 10 years. It is my opinion. So this is our questionnaires. So it uses some kind of branching logic. And in fact, it creates, at the end, a kind of a report for the physician that the physician can look. And this way, since people have already filled up all these questions, they kind of know if they have restless leg syndrome, if they have insomnia and so forth. And it's very important because often these disorders are comorbid. I mean, I told you everyone has a little bit of sleep apnea and insomnia, for example. It's very common that these are associated. And very often people are specialized. They just look for snoring and they forget to ask about insomnia. And what I'm very happy is that you have heard a lot of this genetic talk, beautiful work with Succina at the UK Biobank and also 23andMe, but the problem is I have only 5 questions to summarize the entire sleep field. It doesn't make any sense. So we have actually this instrument that we have developed over a period of years. Now is going to be modified, but they have used a lot of our questions for the UK Biobank, so I think we'll be able to refine the phenotype of all these sleep associations that are subjective with this questionnaire. The second area that I wanted to talk about was the improved PSG analytics. I mean, clearly, machine learning, you all know there's 2 types of machine learning. One that Eileen kind of mentioned a little bit, that's more statistically based machine learning, but also there's this more black box type of machine learning that's called deep machine learning, where you really use neural network and really you try to imitate human tasks. It's called supervised machine learning. And because we have all these annotated sleep studies by technicians that say this is a sleep apnea, this is sleep stage 1, REM sleep, etc. You have these tons of data that's annotated that you can really use with supervised machine learning. And we have done a lot of work. And the way we do that is we use a very large number of sleep studies that are scored, and then we use a neural network that a combination of CNNs as well as RNNs that kind of take into account really the temporal organization of sleep to try to really predict the human labels. And now the way, at the end, we really have developed methods that work better than humans. So you may say, how can you be better than humans? The way we do this is we use multiple humans. So we use, for example, 5 scores that score every sleep study. And then we can create a super scorer that really will be the consensus of each of the scores for each epoch. And then you can show that your machine learning program can actually do better than just predicting the sleep stage of closer, better than each individual score and closer to the consensus. And also thing that's interesting is at the end, you don't get just, oh, it's REM sleep, or it's non-REM sleep, or stage 1, stage 2. You get a probability, a Bayesian probability of whether or not it's this type of stage. And this actually led us to very interesting new hypotheses. For example, when you started to study narcolepsy, we realized very interestingly that certain stages of sleep were not really scoreable. You know, this is a normal sleep study, so the probability of different sleep stages are very clearly distributed. But here you see that in narcolepsy, for example, there's a period of time in the night where the computer doesn't know what to do. It says it's half REM sleep, half awake, half stage 1. It's this kind of dissociated sleep stage, which is exactly what patients with narcolepsy report. They have this weird states where they're half awake, half dreaming, and that's exactly what they report. So you actually see it. So with that in mind, we actually developed another program which used more classic statistics, a little bit like a lasso, that took some of this probability distribution and then used it to try to diagnose narcolepsy. And with many, like more than 900 different patients, we can now diagnose narcolepsy just with a single night of sleep instead of using this MSLT and this SNAP test. So that's just one example how supervised machine learning can actually also eventually start to not only do better than humans, but also get you new hypotheses on how to do things differently and even find new diagnostic procedure. We have also used it to detect very small arousals, when people wake up very briefly, and found that it's better than human. And at the end now, we have pretty much done everything that a classic sleep study does. But of course, with machine learning, we can have a lot more detailed. We can have probabilities of different events. For example, even for sleep disorder breathing, we're starting to have probability of each event as they more like central or more like obstructive, which may again give us other ideas on how to treat these phenotypes in relation to your talk. So, and finally, why do I do that? In fact, I really didn't do that because I'm interested in limiting scores. I did that because I want to apply this in large genetic studies. But before that, I think another area that I just want to mention very briefly is that we definitely need to get rid of the PSG. And a lot of people have been talking about what kind of sensors you need to try to replace the PSGs. And I think actually it should be quite easy. Probably with sound, we would have a lot of the sleep apnea information, and probably with a leg actigraph, we can probably replace the PLMs. And then, but the EEG, I think, will be irreplaceable. But there's definitely people working on this. So, of course, my whole point is, in general, you really need genetics, because why I'm so enamored with genetics, it's like the dogs which I started to study, is because genetics goes to the cause of things. Like, for example, as showed by Anna, if you didn't know what the cause of narcolepsy is, you would just do a GWAS, and you see that they're all immune genes, so you would know that it's autoimmune. However, you wouldn't find the hypocretin, but still, it will really tell you that it's an autoimmune disease. So, as you heard also from Suksena, there's all these very interesting studies that are done in very large amount of people, and they are very powerful in complement to single gene study like Louis Pacek, where you kind of target genes and you find these associations that give you something completely new and unbiased about a phenotype. That's the beauty of genetics. The beauty of genetics is it can uncover completely new mechanism. You don't have any a priori hypothesis. And the second very exciting area is it's always causal, because if you measure something in the blood, it can change with sleep, and you never know if the change in the blood is causing the sleep change. But for genes, you can never change your genes. So as we heard with this concept of Mendelian randomization, it's always causal, and you can actually create this causality pathway across different phenotypes. The problem of these studies, they're very exciting, and we're getting a lot of very interesting data. For example, the couple of things that are really emerging is there's tight relationships with neuropsychiatry. In particular, people who report sleeping too long, they have a lot of genes that they share with schizophrenia and bipolar disorder. Why, we don't know, but that's just a completely new hypothesis that suggests that maybe the way we define some of those disorders it has to be reassessed. Then another thing that's very interesting is we heard is insomnia and RLS seems to share a lot of things. And there's a lot of debate whether or not it's more like a contamination, that people that have RLS complain of insomnia, and that's why you have all this association with these RLS genes, because they have these motor disturbances that manifest as insomnia, and since it was not ascertained, it's a mix of two diseases, insomnia and RLS. But there is also the possibility, very well, that it's not so simple, because, for example, people with RLS, they often, there's a big difference in sex, for example, where the leg movements that are associated with RLS, there's no difference in sex. So there might be, I think, more an interaction. It may not be so simple. And finally, we heard a very nice talk as well about the role of the motor system in sleep. And I think the idea is that most animals, like primitive animals, when they're awake, they move. That's all what they do. They don't sit around thinking, oh my God, what I'm going to talk about tomorrow, like us. So clearly movement and sleep and wake are totally linked in most animals. So it would make sense that there is a lot of wake-promoting systems that are also locomotor systems. So I think maybe this differentiation between insomnia may have a cause also in motor hyperactivation. That's what I wanted to say. I will just give you a couple of examples that suggest that we really need to better phenotype in large-scale study. So one of them that's very interesting, we worked on this disorder for 20 years. I'm kind of a squirrel. I keep this sample for a long time. It's a very odd disorder. People called Kleine-Levin. People sleep for like 2 weeks in a row. They sleep all the time, 20, 25 hours, and they just don't, they don't wake up. And if you try to wake them up, they're very aggressive. They're really weird. I mean, they're not just sleeping. And sometimes they're sexually disinhibited. Sometimes they also eat very odd food. It's a very rare syndrome. It was reported to be maybe more common in the Jewish population, but this didn't pan out. I think it's because it's called Kleine-Levin Syndrome. I think it's a little bit, obviously, I think there's a bias in ascertainment in Israel, for example, where we have a lot of cases. But still, we collected samples all across the world, again, as collaboration. And what we found when we did a GWAS was very striking, is we found this association with this particular gene called TRNK1, which is one of the top associations in bipolar disorder, and a little bit less important in schizophrenia. But of course, the association in Kleine-Levin is much stronger. I mean, the odd ratio, like the strength of the effect is like 1.5, which is much bigger than what you get in schizophrenia or bipolar, where it's only 1.08 or 1.2. That's why we could detect it with a relatively small sample. So maybe some of the phenotype here maybe some of the phenotype in bipolar, are like Kleine-Levin. The other thing we had learned about Kleine-Levin is some patients react to lithium. So we really had an idea that maybe it was related. And finally, when we looked at Kleine-Levin, we had found something very interesting, is that a lot of patients were reporting birth difficulty, like premature labor, all kind of problems like that. And long labor, anoxia during birth, etc., which has also been reported in schizophrenia and bipolar disorder. There's a strong association with birth difficulties. So that kind of fit very well with the idea that Kleine-Levin was maybe a subset of bipolar, but maybe more pure because there is this very strong trunk association. And then I sent a paper and it was rejected immediately because you have to reproduce. So I say, my God, I have 600 cases. How can I reproduce? This is 20 years of work to get these 600 cases. I still figure out to get 200 cases more. I replicate, no effect. So of course, I was ready to kill myself and say, I'm a really bad scientist. I should probably retire immediately, etc. And then 2 months or 3 months later, a big study in Nature came with 28,000 bipolar subjects. And the TRNK1 is the top association. And they do 6,000 more, no effect in TRNK1. And then they explain in the discussion that they don't understand, that it's an outlier. Statistically, it's not possible that it not replicate, but they don't know why. Then I had the idea. And then I was really happy for at least 2 days, because it did fit completely. Maybe there was perinatal birth difficulties that interacted with this TRNK1. The idea would be that 20 years ago, perinatal deaths were much higher. They have been decreasing tremendously. If you look, perinatal care is much, much better now. So in fact, we didn't do anything than simply, stratify by birth year. That was like 2 minutes an analysis. And then Aditya came back and said, yes, that really works. And there were a very, very clear pattern where the odd ratio is very high before the 2000, and then it decreases linearly. So the old people, the ones that were born in the 1980s, have the highest odd ratio because, of course, perinatal care was not very good. And then we replicated it again in our next sample, a new completely independent sample. We separated the people with and with birth difficulty, and we found that this strong point exists only in these cases. So another example of these overlapping traits is insomnia and RLS, which I mentioned. And I think one of the ways definitely to study this MACE1 association is to study this PLMI, for example. So what I would like to claim is that I think we really need to create this resource where we would have both subjective sleep data that we have, genetic data, as well as proteomics, which I would argue later and that I think Aditya will talk about, measure better the phenotype, and really have a relatively large sample, maybe 30,000 people, which should not be too difficult, to be able to do the link between the genetics, the GWAS, and the real phenotype of objective sleep and subjective sleep. And so I think if we do that, I believe that we will be able to have a much better understanding of sleep. And I hope also that it will really create a completely different sleep clinic. We can even use blood biomarkers to diagnose certain disease. That's actually working with circadian rhythm, for example, and also at-home monitoring of sleep disorders. Thank you. These are the participants to the study, in particular Anna and Aditya, which will talk soon, and Eileen as well, who is here.
Questions?
Emmanuel, thank you. Great talk. You just said blood biomarkers of circadian rhythm disorders. So I mean, it's very frustrating that we don't have better circadian measures in our clinical studies.
So Aditya is going to talk about it. I think we have a method that works as well as DMLO, but you can take a blood sample at any time during the daytime. So I think that problem is pretty much solved. People have tried to measure gene expression, but gene expression in white blood cells is not very stable because there are a lot of different subsets. So you always need 2 samples to predict. But with this method of proteomics, we can measure 5,000 different proteins, and we can predict, circadian phase within 1 hour. So I think that problem is solved. And I actually believe that we will eventually find also biomarkers of sleep debt. We'll be able to even predict how much sleep-deprived people are. But that we haven't done the full work.
Could you tell us, what is that TRNK1 gene then? Is it associated with problems?
That's actually very interesting. I have no clue. Because first I should mention it's not TRNK1, because this particular SNP, and I think we heard one of the problems of this association, you cannot never be sure this is causal, and this particular SNP is associated, is a very gene-rich region, and when you look at the change of expression, what's called EQTL, that are associated with that SNP, there are many genes or regions that are modulated, so we don't know first which gene is involved, and the second problem is we don't know exactly how this burst difficulty works. I mean, probably when you have anoxia, if you have this polymorphism, it produces some dysregulation that later manifests as bipolar, schizophrenia, or Kleine-Levin, but we don't understand that. But it's certainly a very exciting finding. And this kind of findings that are called gene-environmental interaction are quite rare. So I think we were very lucky to find it, yeah. But we don't know the mechanisms. That's a big problem of this. The next question is to try to find maybe allelic series in one of these genes with single mutation or polymorphisms that would be associated, and that would pinpoint to a gene, and then after study the gene. So it's all kind. I mean, it's the same as what has been used in schizophrenia. It's like long labor, hypoxia, prematurity, low birth rate, weight. There's a whole list and a scale that has been used, and we kind of follow the same scale. So that too, we should study which subtype exactly associated, but it's a small sample. Sorry.
Lovely talk, Emmanuel. What's your thoughts on, I agree with the shift towards the way we monitor. At the moment, clinical decisions are made on a single study often.
Yeah.
What are your thoughts about night-to-night variability and measuring over a longer period of time, and how technology might help us with that? I mean, I'm thinking about the recent data showing that, they measured sleep apnea over a month, and showing that in people with AFib, and the worse the sleep apnea the night before than the AFib worse the next day.
It's amazing, but when I started in this field, I mean, even for narcolepsy, the MSLT was the test, the gold standard for the test, and I realized that it had only been tested in 10 controls, in the 1980s, and it was used clinically in millions of people, thousands of people every year. I think the same way, we don't really know the night-to-night variability. And I totally agree with you, especially we overestimate the HI because we force people to sleep on the back. So we really need an at-home method that's better and comfortable. And there's no doubt that it will happen the next few years. So big question, and I hope you will discuss that, is which sensor do you need? Do you need a bed that's measuring everything without touching you? Do you need I believe you need brain activity because I'm kind of biased, but I do think that we have to simplify it. And I think sound is a very powerful method, but may not be the only one. And I mean, we are working with Stanley and to also maybe do sleep surgery and try to look at the pattern of collapse in relation to the snoring, which I think could maybe help some of the issues that you're discussing about different phenotype. It's a long story.